This is the title slide. The title was advertised was the NSSR is a logical place. NSRR is a logical place to deposit animal-based sleep recordings. And I'm going to use one data of the two data sets that Michael mentioned that we've donated to the NSSR as an example. So just to give you an idea where we're going, I'll introduce first the nociceptin orphan FQ NOP receptor system, talk about how there's interaction with that with the hypocretin system, and then talk about some pharmacology studies that were conducted in 3 different species that comprised a portion of the paper we published in PNAS earlier this spring. Due to time constraints, I won't get into the mechanism of this other than reporting the pharmacology, and then jump to why I think NSRR is the best place to deposit animal-based sleep recordings. So with that as background or introduction, to introduce the NOP nociceptin orphan FQ NOP receptor system, I just want to remind you that this receptor was originally cloned in 1994, and so nearly 30 years ago. And it got a lot of interest in neuroscience generally because there was a lot of sequence and structural homology with opiate receptors, but the pharmacology of the system was distinct from classic opiate receptors. And subsequent to the year subsequent to identification of the receptor was deorphanized, and the endogenous ligand was identified as this 17 amino acid neuropeptide that was alternately called nociceptin by one group and orphan FQ by another group. And the sequence of the nociceptin orphan FQ has high homology with another opiate molecule, dynorphin A. Since the description of the system has been implicated in a number of different functions, including pain, drug abuse, and stress-induced analgesia, studies that we conducted 15 years ago or so. And because of the distinct need for non-addicting, pain-relieving medications, there's been a lot of interest in a focus on this system, particularly on mixed mu NOP agonists, because these NOP does not seem to have, activation of the NOP receptor does not seem to be addictive. So about 15 years ago, as I mentioned, we published this paper in Journal of Clinical Investigation, where we looked at the effect of nociceptin, which is what I will call this molecule, on the hypocretin system. At that time, we were studying various inputs to the hypocretin or orexin cells. And one of the tools we used was the orexin GFP mouse, which allowed pathological electrophysiology studies. As indicated here at both the light and EM level, we showed that nociceptin directly innervates the hypocretin neurons, and that when we applied this neuropeptide to a slice containing the hypocretin cells and recorded from those cells, we saw that nociceptin immediately reduced firing activity, and the effect was direct, as indicated by the presence of TTX. Now, and the effect was blocked by the nociceptin or NOP receptor antagonist, SR14148. We also, although I won't present this data, we also showed in the study that the function of this input probably has something to do with relieving stress-induced analgesia. In the course of doing those studies, we noticed that some of the animals injected with the nociceptin peptide became sleepy. So that was the genesis that led to this paper that was published in PNAS earlier this year that I'll describe in the next few slides. We conducted a number of different species studies in, as I said, in 3 different species: rats, mice, and non-human primates. For the rat studies, we were conducted in Sprague Dawley rats. The animals were implanted with DSI telemeters that allowed measurement of EEG, EMG, and body temperature and locomotor activity. We conducted 2 studies that were basically a repeated measures design in which each of the animals received 8 treatments, a vehicle treatment, 3 doses of a NOP agonist known as synthesized by Roche called 646198, and zolpidem as a positive control. And then a second study with a different compound synthesized at SRI that was also a NOP agonist, again, 3 doses in zolpidem. We recorded EEG and EMG for 30 hours, 12 hours before a baseline period, and then an 18-hour post-injection. The EMG was manually scored in 10-second epochs for 6 hours before injection and 18 hours post-dosing. We measured parameters such as sleep latency, the hourly amounts of state, and EEG spectral analysis, analyzed the data by ANOVA. So this this slide depicts the 2 molecules, the Roche compound on the left and the SRI compound on the right. And you see that both of these have high affinity for the NOP receptor and a modest affinity for the mu opioid receptor. That's what that symbolizes here, MOPR is mu opioid receptor. With both of these compounds, you will notice that there is a dose-dependent reduction in non-REM latency and a dose-dependent increase in REM latency. And the reason REM is extended, you will see, is because there is such a profound effect on non-REM sleep. So you'll be seeing a series of slides that have this format, where on the left we show the hourly distribution of the, in this case, wake time, under the 5 dosing conditions, in this case with the Roche compound 6198. And on the right, you'll see the data binned in 6-hour bins. Again, for each bin will have those 5 dosing conditions. And what's evident, the arrow here indicates the time of injection in which the animals received oral administration, not injection, of 6198. And as you can see, there seems to be a dose-dependent reduction in wake time, as summed here over a 6-hour period. And the reduction at the highest dose of wakefulness is significantly different than the amount of wakefulness reduced by treatment with zolpidem. In the subsequent light period, there does not seem to be any rebound sleep, except in the case of the animals treated with, rebound wakefulness, except in the animals treated with zolpidem. This shows the converse data, the increase in non-REM sleep that occurs in the 6 hours subsequent to injection, and therefore you see this dose-dependent increase in non-REM sleep relative to zolpidem. And again, there seems to be no increase rebound sleep, except perhaps in this lowest dose of the NOP agonist for the first hour or 2. The mechanism, of course, when one looks at an increase in sleep in a rodent on an hourly basis, there's 2 possibilities. One is that you could have longer bouts of sleep during that hour, or secondly, you could have more bouts of sleep during that hour. And of course, if you have more bouts of sleep, that's indicative of some evidence, provides some evidence of sleep fragmentation. But as you can see from this graph, the animals in the hour, the rats in the hour after injection were almost continuously awake for the first hour, and certainly profoundly awake in the second hour. The inset here just shows the increase relative to zolpidem. This on the right shows the number of non-REM bouts, and of course, if there's an increase in non-REM bout duration, of course, there has to be a reduction in the number of bouts during that hour. This graph shows the distribution of REM sleep time, and as you can see, for the first 5 to 6 hours after injection, there's almost complete elimination of non-REM sleep that only gradually recovers during the subsequent light phase. Here shows the dose-dependent decrease in non-REM sleep, or at least the profound decrease in REM sleep at the highest dose, and that continues into the first 6 hours, at least, of the light period. And I want to note that this decrease is only occurring at the highest dose of 10 milligrams per kilogram. We showed very similar results with the other SRI NOP agonist, 16835, where you see the dose-dependent reduction in non-REM sleep, in wakefulness, an increase in non-REM sleep, and again, a profound elimination of REM sleep that continues into the light phase. So both of these compounds were producing very similar results in the rat. We also measured body temperature and locomotor activity in response to these administration of these compounds. And of course, if the animals are sleeping more, there's going to be a reduction in locomotor activity, as evidenced by the asterisk above. But what's also revealed by the body temperature measurement, and this is intraperitoneally measured body temperature, is that there is a profound decrease in body temperature on the order of 1.5 degrees with the highest dose of the Roche compound, and a less profound decrease with the SRI compound. But again, it's only at the highest dose that you see this reduction of body temperature. But I would note that it's within the range that one sees across the 24-hour cycle, at least with the SRI compound. Perhaps the NOP compound from Roche was more potent in that regard. In conjunction with the increase of non-REM sleep, there was a dose-dependent increase in non-REM delta power, with the highest dose producing the highest increase in non-REM delta power with both of these compounds. So given these consistent results with two different compounds in the rat, we then wanted to evaluate whether the compounds, first of all, were effective in other species, but secondly, also convince ourselves that these compounds are working through the NOP receptor. So for that, we undertook a second study in mice of a very similar design, but in this case, we had access to the OPRL1 knockout mouse. That's the gene symbol for the NOP receptor. And we had access to a limited number of mice, but 6 heterozygotes and 5 homozygous mice. They were 7 to 10 months of age. They were planted, and in this case, we only looked at the more potent compound, the Roche compound 646198. Again, at the same 3 doses, indexed it against zolpidem, and scored EEG and analyzed it in a similar manner. Although we analyzed it in a similar manner, I'm going to present here cumulative changes over a 6-hour period post-dosing. Again, dosing was occurring in the middle of the dark phase, the animal's major active period at ZT19. Vehicle is in black, and as you can see, there is a dose-dependent reduction in wakefulness time in the wild-type mouse. That effect is attenuated in the heterozygous and completely eliminated in the knockout mouse. The zolpidem effect, zolpidem is still effective, of course, in the knockout mouse. Conversely, when we looked at non-REM time, there's the dose-dependent increase in non-REM, as I mentioned in the rat. Again, attenuation in the heterozygous and a complete elimination in the knockout mouse. Consequently, we're convinced that this compound is affecting sleep through the NOP receptor. So the third study we undertook then was to look at an organism that is diurnal, and of course, one that is closer to humans than the rodents. And for that, we had access to 5 cynomolgus macaques. Again, we used DSI transmitters to measure EEG, EMG, and activity. We only used the Roche compound in this case, and I'm only going to present data from 1 dose. And that, in contrast to the previous studies, the animals were administered this compound just before lights offset, so 15 minutes before light offset. So we're looking at the effect of the compound across the night. We scored EEG using parameters similar to the way what sleep is scored in humans, and analyze data similar to the manner I told you before. So first of all, in terms of sleep latency, you can see this dose of 646198 significantly reduced non-REM latency, and there was a trend towards an increase in REM latency. If I direct your attention first to the graphs down below, this shows the hourly distribution of wakefulness, non-REM, and REM time across the 12-hour dark period. Here, the arrow here indicates the time of dosing, and the Roche compound clearly is reducing the amount of wakefulness, increasing the amount of non-REM sleep, and there is an initial suppression of REM sleep that lasts about 6 hours before it starts coming back. Although I've talked here about non-REM sleep, of course, we did break the non-REM sleep into N1, N2, and N3 according to the criteria used for scoring human sleep. But to make the data comparable to what we saw in the rodents, we combined the REM sleep here, the non-REM sleep here. I guess the most notable difference is that in the first half of the night, here is ET3, 13 to 18, there's an increase in non-REM in N3 sleep, whereas in the second half of the night, there's no significant change in N3, but there is a slight increase in N2 sleep. Again, you see the suppression of REM sleep across the first half of the night, but the recovery during the second half. We also conducted spectral analysis, and clearly so the upper panels here are the vehicle, the lower panel are 3 derivatives of animals injected with the NOP agonist, and as you can clearly see, there's an increase in delta frequencies relative to the vehicle injected animal. So all the data that I've talked about from the primates here are available at sleepdata.org, at this website. So let's now bridge to discussing why you want to share your recordings at all. Well, I guess it's fair to say there's more sticks than there are carrots at the moment in order to doing that. There's certainly a mandate from the NIH. And depositing your data allows you to then fulfill that mandate. There are journals that require this. Our paper would not be accepted into PNAS until we had deposited the data. Of course, in most cases, not all, but most cases, we're talking about data that's been collected with taxpayer funds. So there's a good rationale for making it generally available so that the scientific community as a whole could benefit. And I think, as Michael alluded to in the beginning of his introduction, certainly there are every laboratory has its limitations in terms of the way it analyzes its own data that it collects. But there are other individuals who evolve new techniques for analyzing data over time that may provide unique insights to the data that you've collected. And I think all of us want to see that our contributions to literature get exploited by others going forward. Now, there are a number of neuroscience-oriented databases out there, OpenNeuro, NEMO, for example. These are particularly well-suited for imaging data, both from human and microscopy data, and a number of other NIH HEAL recommended repositories. But I've chosen to deposit these data at the NSRR for a number of reasons. One of which, as Michael alluded to, it's been NHLBI funded, and the work that I've talked about to this point was funded from NHLBI through the HEAL initiative. The NSRR was founded in 2014, so there's some evidence of stability. If you look on the website, or those of you who are not familiar with it, you'll see that there are data sets, there are tools that are available for analysis, and there's an effort towards data harmonization, which of course is a much larger problem in the animal data collection area, animal-based research than in humans. And there at NSRR also provides webinars and community events such as this. So what are the steps involved in depositing your data? Well, first you have to complete an initial study intake form It can be found here as an example of what it looks like, and it's just how they how the data were collected, how they can contact you, what the data, the size of the data is gonna look like, and so forth. You then compile your documentation. That produces essentially a README file that allows others who access your data to understand how it was collected. And here's what the README file looks like, or at least a portion of it, the web page in NSRR for the data collected in non-human primates in the study that I just described. In some cases, you'll have to obtain a release from your institution to allow the data to be deposited. In conjunction with NSRR, you'll prepare the final data sets with any dictionaries and descriptors that are necessary, and then upload your files through the NSR through a secured FTP site. So let's not neglect the fact that all these steps take time, and particularly when you're at the last stages of publishing a paper. This ends up being a time sink that, at least to this point, we have not had to deal with. But going forward, in order to benefit this scientific community as a whole, these are the types of things that we'll have to be engaged in. And I think the interaction that I've had with NSRR in terms of depositing our data has not only been very simple, but also they've been very enthusiastic about accepting our data. So I'm very happy about that. So just to summarize the scientific portion of what I presented, and as I said, I did not get into the mechanistic aspects. Everything I presented was really pharmacology data. But anyway, NOP agonism reduces latency to non-REM sleep in rodents and non-human primates. There's dose-dependent reduction in wakefulness in non-REM sleep as well. The EEG delta power is increased by NOP activation in both types of species. But at the highest doses of NOP agonism, REM sleep is suppressed and body temperature is reduced. So we probably exceeded perhaps the highest dose that would be useful, at least in these animal studies. And what I'm ultimately leading to is that the nociceptin NOP receptor neuropeptide system may have a previously unrecognized role in sleep-wake regulation. And I just want to acknowledge my colleagues who've contributed to these studies over the years. Steve Moriarty and Yu Sun in particular were co-first authors on the paper. And we've had generous support from NIH over the course of the study. And these are other folks who've contributed either resources or tools that some of which are unpublished at this point. Thanks for your attention.